and I would like to thank the host, the Indian Photobiology Society, for inviting me. And especially, I'm excited because I have worked on at the Bose Institute, at the museum, so I am a little bit aware of the kind of work that Jagadish Chandra Bose did. Often his work is not misunderstood or incompletely understood. And what I want to say, because this is the Photobiology Society, I want to talk very briefly about Jagadish Chandra Bose's contribution as a pioneer in science. But I want to focus on his work in photosynthesis research, which is not understood or known so well. I'd like to dedicate this lecture to our previous director, Professor B.B. Bishwash, who, if I understand correctly, was also involved very much with the Photobiology Society. And it's an honor to be with this society, because when we were students at Presidency College, we read books, the photochemistry book by Professor Rohati Mukherjee, and as I understand, she was one of the founding members of this society. So I'm going to start by a slide. I'll do the impossible. I'll try to consolidate all work by Jagadish Chandra Bose in one slide, and then I'll follow it with his work on photosynthesis. So Jagadish Chandra Bose was born 1858, 3 years before Rabindranath was born, about 5-6 years before Vivekananda was born, and he died in 1937. The first 10 years of his life was spent in Bangladesh, where he, present-day Bangladesh, undivided India, where he was born. And it is extremely important to recognize that he spent his childhood, 10 years, in a very rural setting. By rural setting, I mean he had direct interaction with nature. He went to a Bengali medium school. He played with children who were farmers' children, who were fishermen's children. And this is extremely important, because I think later on we will see that even though he did physics, he moved away from physics, because I think he was an accidental physicist. He was truly a naturalist. He was interested in understanding nature. When he was about 10 years old, 12 years old, he came to Kolkata and he joined St. Xavier's College, school and college. And there his mentor was Father Lafont. He was a Belgian priest. He was heavily involved in starting Indian Association for Cultivation of Science with Mohanlal Sarkar. And it is under Father Lafont's guidance at St. Xavier's that Jagadish Chandra Bose became interested in physics. But interestingly, when he finished his college at St. Xavier's, he went to England not to study physics, but he went to England to study medicine in London from 1880 to 1881. But he quit. He quit because of ill health. So this was also an accident. If he did not have ill health, he probably would have become a doctor. He then moved to the Christ College in Cambridge and did a degree in natural science. So there he had biology, physics, chemistry. His physics teacher was Lord Rayleigh, that we heard about just before. Darwin's son taught him. Sidney Vines taught him biology. So he became a naturalist, basically natural science. And he returned to India after that, and he joined Presidency College. I'm just giving an overall view so that we understand where his research really lies and where does photosynthesis lie. Presidency College he joined in 1884. He married Obala Bose, an eminent feminist who worked hard for women's education, in 1887. And many people know, but in case you don't know, when he joined Presidency College, he did not get salary as much as the white men got. So he, as a protest, stopped taking salary, but he worked full time. And in 1888, he got his full salary. So Jagadish Chandra Bose can be considered to be the first Shotagrohi person. Before Mahatma Gandhi, who came back to India from South Africa only in 1915. Interestingly, until that time, we don't hear Jagadish Chandra Bose doing any research. At the very beginning, from 1888 to 1894, almost 6 years, he pursued what is called his scientific hobbies. They included photography. He was one of the earliest photographers in India, and X-ray. For X-ray, he also worked with Nilratan Sarkar, the famous doctor, and produced some interesting work that I will not discuss here. And it was in 1894 that he started his core research work on microwave. And he did it only for 5-6 years. That was his first phase of research. He pioneered in generation, detection, and propagation of millimeter waves. I will not elaborate on this. I can give a 1 hour, 2 hour lecture on this to show what he did, how incredibly genius he was, how much of a pioneer he was. Interestingly, around 1899 to 1902, he shifted his research from microwave research to living, non-living. And what he was trying to do was he was trying to give an experimental proof that there is no boundary between living and non-living. And the natural transition started because he was working with detectors when he was working with microwave research. And therefore, any detector, a physical detector, ultimately is how a material responds to external stimuli. And so he started giving stimuli to living, non-living, getting electrical signals. He moved away from this. And he started his long journey in plant physiology from almost 1903 to a little bit 4 or 5 years before his death in 1933. And there, one of the main findings was, as opposed to chemical signaling that was understood then, he showed that electric responsive plants. He pioneered in plant electrophysiology, photosynthesis. He designed and built novel devices. By the way, he didn't have to import any instrument from outside. He built them all, designed them, not 1, not 2, not 3, not 10, in hundreds. And subtle plant responses to external stimuli. So and then he started Bose Institute after he retired from Presidency College in 1915. In 1917, so remember, in 1917, he was 59 or 60-year-old. That is the time that typically in national institutes people retire. So he retired and started a brand new institute. And if you look at his journey, scientific journey, I just want to add, just to give you the dimension, that during this entire period, he had made many visits to Europe and America and Japan, and also actually spent 1 year working in Davy Laboratory in England. So 1896, he went to UK, France, Germany at the Royal Institute. That is where he first got his recognition demonstrating his microwave device. 1900 to 1902, he went to Paris. He made Vivekananda in Paris. He gave lectures at Royal Institute and spent working in Davy Faraday Laboratory for 1 year, and actually contemplated staying back there because he was offered professorship there. 1907, UK, USA. Again, 1914, Royal Institute, USA, Japan. 1920, Europe. That's when he became a fellow of the Royal Society. 1923, he again went to Prague, UK, Denmark, became very close to George Bernard Shaw. 1926, he met Einstein. At that time in Europe, he was working for the League of Nations. Einstein was part of it. 1927, France. 1928, Europe, Egypt, League of Nations, and Europe again. This is just to give you a summary. And what I want to show you here, 2 points I want to make. An important person in Jagadish Bose's life who influenced his scientific research is Sister Nivedita. Sister Nivedita came to India in 1898 and she died in 1911. So this is the period where Jagadish Basu overlapped with Sister Nivedita. So you have to understand Jagadish Bose had a research career before Sister Nivedita, which was purely microwave, with Sister Nivedita, which was living, non-living, and after Nivedita, he moved away from living, non-living and became into plant physiology. I want to point this out because it's the period of living, non-living work that he stopped publishing in peer-reviewed journals simply because peer-reviewed journals would not take his papers. And so he decided, with support from Sister Nivedita, that he's not going to send his papers to scientific journals for peer review, but publish his results in books. And we will see the book on photosynthesis that he published in 1924. They're all papers in a book. And what I'm going to do now, I'm going to focus on photosynthesis, his research on photosynthesis. Not much is known. Not many people know what he did. I'm going to point out to you a single author paper that Jagadish Bose published in Nature in 1923. We saw a paper by Raman, by our previous speaker, single author paper, 1921 it was. In 1923, he published a paper in Nature from Bose Institute called Effect of Infinitesimal Traces of Chemical Substance on Photosynthesis. Remember, this is 100 years ago from now. And we are talking about a man who was 65 years old then. So I just want to understand, recognize that we are talking about research done by a 65-year-old man. I'm not 65 yet. And this was done 100 years ago. If you look at the thing, at the abstract, he says that he has made a new device. And he says that it is too little space to say what he has done. He is publishing a book soon called The Physiology of Photosynthesis, and it is all there, all. And then we look into the now, let us, what did he do? What did Jagadish Basu do in photosynthesis? There's a whole book with almost more than 20 chapters that he dedicated in this book. The book is called The Physiology of Photosynthesis. It was published by Longman Green, and it has a single author, Jagadish Chandra Bose. I must add that Jagadish Chandra Bose was notorious for publishing single author papers. I think Prafulla Chandra Rai once told him that, why don't you give credit to others? But, you know, everybody is unique. And so in the preface of this book, Jagadish Bose actually acknowledges two people. So if he did publish the paper today, he would write the name of Professor N.C. Nag, who was assistant director there, and scholars of Bose Institute. So let us delve into this book, which was published in 1924. At the very beginning, the introduction, he introduces photosynthesis as was known then. What was known about photosynthesis then? So he describes what is already known about photosynthesis. Then he justifies why is he doing this work. And I want to say, he says that it would appear, therefore, that almost everything that can be known about photosynthesis has now been ascertained. It may be admitted that this is approximately true in qualitative sense, but not in the quantitative sense. So here comes Jagadish Bose. He's one of the first person to take biology from a qualitative subject to a quantitative subject. Then he says that what he has done in this present volume is essentially a record of quantitative research in various directions. So let us remind ourselves, he uses the word qualitative sense, it is known, quantitative sense, it is not known, and he itemizes these two words. And then he said, this is what he has done. What I'm going to do is, in this short talk, I'm going to take you through the book. I'm going to summarize chapters, and I'm going to skip chapters that are not so relevant. I'm going to point out things that he did that was pioneering. So let us keep this in mind. Quantitative is the key. And very understandably, he is considered to be the first biophysicist, quantifying biology from a physicist viewpoint. So chapter 1 was the introduction. Chapter 2 says evolution of pure oxygen under light. What he does there, that he describes in detail how to remove all gases except oxygen that he is supposed to measure, which is a product of photosynthesis. And then he introduces a plant, a water plant called hydrilla, that he's going to work on. Second chapter is called determination of rate of evolution of equal volumes of oxygen. This is where he describes his instrument. It's a vessel. Inside the vessel, you have the plant, hydrilla. If you shine light on hydrilla, the hydrilla light is going to produce bubbles of oxygen, but the bubbles are of unequal volume. So you cannot count the number of bubbles because they do not have the same volume. So the idea of equal volumes of oxygen. So what he said is that I am going to allow the bubbles to go up. There's a U tube here. On the top, there is a little bit of mercury. It's going to push the liquid in the U tube, some viscous oil, and it's going to push the U tube. And then when there is enough pressure, the material in the U tube is going to go up and the bubble will get released. That means every time it gets released, the air gets released, it's equal amount of oxygen. And chapter 4, it says he automates his record. What he does, he puts a little electrode on the top right here. On the top is mercury. So every time the bubble, the entire air escapes, a circuit is completed, and as the mercury on the top touches the metal tip, and the gas escapes. That means if you do an experiment, this is the device that he used. It's extremely important because then he proceeds to do a lot of experiments with this. Here is this bubbler. Here is the mercury on the top of the meniscus. Here is the electric pen, E. And every time mercury goes up and touches the pen, what you see is you see a dot here. This is moving, this cylinder is moving with uniform rate. And every time, the circuit is completed, it touches this and you get a dot. So what you see, his results is on the top. So you see dots, 1 dot, 2 dot, 3 dot. And what you see is the top here, it is under low light. That means every time there is dot, so your x-axis is time. Every time you see a dot, that means the air has escaped. Equal volumes of air has escaped. At low light, it takes long time for the air to escape. In medium light, the time decreases. At strong light, the time decreases further. So how often the dot is appearing as a function of time is actually a quantitative measurement of the rate of photosynthesis. So he actually devises a quantitative way of measuring the rate of photosynthesis. I think he was the first. Chapter 5, he shows and measures for the first time photosynthesis under increasing intensity of light. So on x-axis is the intensity of light, y-axis is the rate of photosynthesis, and the top curve is sunlight, is summer sunlight, the bottom is winter. And such curves, I think he was the first person to generate such curves, non-linear curves, saturation initially increasing linearly and then saturating. Chapter 6, called relation between the quantity of light and the amount of photosynthesis. The same curve, but the top curve is with the sunlight and the bottom curve with an artificial light, because at that time, it's a tungsten arc lamp that was available right then. I'm going to skip this. I'm going to come to chapter 7, the physiological factor in photosynthesis. For example, this is again, he measures other things with light, like phototropic curve. Phototropism is movement, physical movement of a plant, not photosynthesis, say towards the light. If you take a plant and put it away from the window, the plant will turn towards light where the window is. Again, I'm not going to go into details here, but these are the kind of quantitative measurements that he was doing. Then next chapter, he goes into changing photosynthetic activity under stimulus, anesthetics, and poisons. So he devises something else where he can add through this funnel anything that he wants to. Here, what you see is he's measuring on the y-axis photosynthetic activity. On the x-axis is how long they're applying ether as an anesthetic. So 0 minutes, 0 second, so 0 minute, 200 minute, 400 minute, etc. And he could actually measure quantitatively the effect. I'm going to go to the next chapter. the effect of infinitesimal traces of chemical substances on photosynthesis. This is the work that he published in Nature. It's very interesting. I want you to think of him as that naturalist who spent his childhood in a rural Bengal, today current Faridpur in Bangladesh. And this is what he writes in his chapter. My attention was drawn to the matter by the extraordinary great increase in the photosynthetic activity of hydrilla after 3 days of thunderstorm and rain, which lasted from the 10th to the 13th February of this year. In 1923, there was a little pond inside Bose Institute's Rajabazar campus, and there was a lot of storm. And what he saw was, wow, the photosynthetic rate has increased. So what he did is he hypothesized that there must be acid rain, there must be nitric acid coming from the rain. Maybe small amounts of nitric acid is responsible for increasing photosynthesis. So he used his device, and on the y-axis is the concentration, x-axis, y-axis is the photosynthesis activity. Just look at the top curve, don't worry about the other formaldehyde or thyroid. What he showed is at very low concentrations, very low, if you add nitric acid, the photosynthesis rate goes up. It reaches a peak, then it slowly goes down. And I don't think he was very clear why this was happening, but he repeated it. And this was something that he wanted to explore. It doesn't matter what you observed, whether he understood it or not, but this was quantitatively measured. I tried to find out what it means from today's understanding of science, but with my little bit of try, I could not. I'm going to skip this, come to the next chapter. The electric response to light. Remember, he was working with electric response. So again, he devised something new. Here is his plan. He gives electric response. Again, I'm not going to explain this. One is called a metabolic, the other is called anabolic. One shows positive, the other showed negative. But I'm just going to chronologically show where he's leading to. Then he moves to phenomenon of photosynthetic induction, meaning he showed his plant some period of darkness and then showed light. And x-axis is increasing the period of darkness, and then it on the y-axis, how long does it take after a period of darkness for the photosynthesis to start? Again, what it means is secondary, but this quantitative and measurement. So his conclusion was the induction period increases with increasing duration of previous darkness. In a very natural way, in the next chapter, he moves to effects of intermittent light on photosynthesis. So he shows light, stops light, shows light, stops light. This is the period of intermission. And he started seeing very interesting curves, like intermittent illumination of increasing duration lowers photosynthetic efficiency till a turning point is reached. Chapter 13, he designed something else. It's called automatic radiograph. He uses selenium cell. And the idea of automatic radiograph is to measure intensity of light as a function of wavelength. And what he does, he measures photosynthesis during the entire day from morning to night. And he also measures independently the amount of light from this device and temperature through a thermocouple. And so he's now connecting temperature, light, and photosynthesis. So this is actually this data actually can be plotted in 3 dimensions. It's a 3-dimensional data. Next chapter, he builds another instrument. I mean, he is non-stop. He's coming up with instruments and after instruments. This is also for measurement of intensity of light. And this, he says it is portable. So he can take it outside. He can measure sunlight, etc. Next, what he did was something very important. We showed you that he measured the rate of photosynthesis as a function of intensity of light. This is rate of photosynthesis as a function of intensity of carbon dioxide. So it is not linear. We now know this. But he established it quantitatively for the first time. Chapter 16, I have done blue because this is where he did something incredible. He showed that photosynthetic evolution of oxygen happened even when there was no carbon dioxide. So what he showed is that hydrilla, the plant he was working with, stopped photosynthesis in winter in neutral pond water when there was no carbon dioxide. But in summer, when the pond water was acidic, there was photosynthesis even when there was no carbon dioxide. He read, then he realized that maybe the acidic water has something to do. He looked at, and then he recognized malic acid could be present. It may be malic acid, which is the source of carbon, and not carbon dioxide. And so what he did is he did the same curve on the left, one with carbon dioxide and one with no carbon dioxide, but increasing amounts of malic acid. And this is what he found. I just want to point out to you by a pioneer, a very well-known person, an American of Indian origin, Professor Govindji, who is in the University of Illinois at Urbana-Champaign. This is something that he wrote to me in a private communication. What he wrote is the discovery of this, that means photosynthesis without carbon dioxide in presence of malic acid, quite new pathway later turned out to be the C4 pathway, now the well-known Hatch and Slack pathway. So I'm not going into the details, but this is a pioneering work, and in his words, thus Bose was a pioneer of photosynthetic research not only in India but also in the world. If you guys are interested, they wrote a Raghavendra from Hyderabad and Govindji from University of Illinois, Urbana-Champaign. They wrote a chapter, a pioneer on this. This is the book. If you need a copy, you can send me an email. I'll share it with you, but I'll go ahead for lack of time. Chapter 17, we are now. Effect of variation of temperature on photosynthesis. He devised something else. This is another instrument where this is a water bath. The whole thing goes inside the water bath. They maintain temperature. And again, he generates curves that we know now, but was not known then, that with increasing temperature, photosynthesis increases, then it reaches an optimum, and then it comes down. I'll move on. The tonic factor of photosynthesis. So then he looked at effect of season, stimuli, daily variation in photosynthetic activity. So for example, this is another setup that he designed. He called is the complete apparatus, where he can measure light intensity, photosynthesis activity. On the x-axis is time of the day, and the y-axis is activity of photosynthetic activity. And he goes into details that, again, it's not right to go into details here, but I just want to show how he's trying to find out what are the factors that affect photosynthesis. If I change them in a systematic manner, can I quantitatively measure photosynthesis? Chapter 20 is very important. Chapter 20 is called photosynthetic efficiency of light of different colors. In other words, he is trying to probably first time measuring the photosynthetic curve that, like as a function of wavelength, the photosynthetic activity curve. I want to point out, many of you know here, Engelmann in 1882 did a very interesting experiment. He took a microscopic slide that is shown here, and he covered the slide with green alga. Then he took a prism and he projected different colored light across the slide. So there is alga on the slide, then it is illuminated with different kinds of light, and then he put some bacteria. He put some aerotactic bacteria on it. So anywhere oxygen will get evolved due to photosynthesis of the alga, the bacteria will move there. What he saw was the bacteria moved around this area, the red area, and the bacteria moved along blue area. So he was the first in 1882 to say that photosynthesis is effective at two different wavelengths, red and blue. But there were other experiments that they were not very clear what was going on. and there was nothing very quantitative. And so Jagadish Chandra, what he did is he devised another instrument, and what he did is he used a light source, blue, using filters, and red, using filters. In his device, he had a plant that was submerged in the water. And he said that as the plant starts photosynthesizing, it is going to produce oxygen bubbles on its surface, and the plant will get a buoyant force and it will rise up. So he said, I'm going to measure the distance of the plant from the surface, and that will give me a precise information about photosynthesis. So as the submerged plant in this beaker releases oxygen bubbles, it moves towards the surface. Upon reaching the surface, the bubbles burst and the plant submerges again. So he measured the rate of ascent. He also measured periodic flotation frequency. Faster the frequency, faster is the photosynthesis. So high frequency versus low frequency. Then he plotted the rate of ascent. So how quickly the plant is going up. So it's a kinetic experiment. Distance from the top versus time. This is what he measured with red light. This is what he measured for blue light. And so at the top, when it comes down, that means it has reached the top. And so what he said, both red and blue induce photosynthesis. Red light is more efficient, blue light is less efficient. Then he measured the frequency of the plant ascending and coming down, and he found out the relative power. And here I want to show you what you have is the relative efficiency. Y-x-axis your wavelength. The green curve is a very recent curve. It is an average of photosynthetic action spectra for higher plants, shown as the average of 22 crop plants, the green one. And if you integrate the green curve in the wavelength range that Jagadish Chandra works, 400 to 475 nanometer, this is what Jagadish Chandra Bose is, the blue guy. This is the McCree is the green integration at 605 to 760. This is JC Bose, that is McCree. So this is incredible. In fact, he was one of the first to determine the photosynthetic action spectra for higher plants. Then he went to determine the energy of different rays of solar spectrum using a carbon disulfide prism, devised another instrument called magnetic radiometer for measuring intensity of light, where it had a zinc strip that lengthened by the rise of temperature produced by absorbed radiation, which was then magnified by this mirror. And what he measured is light intensity. It's a relative light intensity of sun as a function of wavelength, because he's going to use this later to measure the photosynthetic activity. So then he does determination of the photosynthetic efficiency of the spectral rays. The red guy here is the same red guy here. So this is the energy from the sun at different origin source from different at different wavelengths. And the blue one, the green one, is the photosynthesis efficiency. Now what you see is, since the light falling at different wavelengths are not the same, what I did is I normalized the photosynthetic efficiency with the intensity of light, and the green curve on the right that you see is the photosynthetic activity adjusted to light intensity. And the red curve you see is that average thing for the plants. So what you see is he detected the red peak. He detected the peak at the right wavelength, but he missed the blue peak. There is no peak in his green curve here. Now, I think because in the blue range, his light intensity was very low, and there was not sufficient photosynthesis for him to pick up. Interestingly, if you read the chapter, this is what Jagadish Chandra writes. There is, however, no second maxima at the second absorption band. So he knows it is not there. So he is trying to find out why. Then he says, maybe this light is being absorbed, but for something different, for phototropism. So what he says, this is again what he writes in his paper, in his book. Experiments are described which tend to prove that these blue-violet rays are concerned in inducing phototropic curvature. And today we know that phototropins are primary photoreceptors that control phototropism by absorbing UV blue wavelength. In fact, it is the flavin mononucleotide that absorbs. So in some ways, he was not aware, but Jagadish Chandra was first to identify, wavelengths where phototropism is responsible. Then what he did is he went on. He's non-stop. This is chapter 23. I think 4 PhDs is what this one book. So determination of the increase of weight. So he came up with something called, so every chapter has a new, unique, innovative, instrument, torsion balance. And he measured the weight of the plant as a function of photosynthesis. And he did calculations to find out what is the carbohydrate that is getting stored. And he found out, again, it's a quantitative thing, that the maximum rate of formation of carbohydrate in hydrilla under perpendicular incidence of sunlight is 2.8 gram per hour per square meter. He tested this by a different method, which he describes in next chapter, saying simultaneous determination of carbohydrate formation by 2 independent methods. He comes up with a different method called the chemical balance, and he shows the same thing. So far what he has done, he can measure the intensity of light at any wavelength he wants. He can measure temperature. He can quantitatively measure photosynthetic rate. He can even measure the amount of material, carbohydrate stored as a function of photosynthesis. So he goes, in chapter 25, is called efficiency of the photosynthetic organ storage of solar energy. Today we are interested in solar energy, and when we talk about a solar device, we always say, what is your efficiency? That means the number of photons that are the material is absorbing, how much of it is actually being translated into energy. So there are photosynthetic efficiency of plants. And what he says is that when he was working, it was very difficult to measure. It was not very clear. So what he said is that I am going to measure the photosynthetic efficiency of hydrilla. Again, it's an apparatus for determining the photosynthetic efficiency. There is he even used a calorimeter to measure the heat. I don't want to go into details. We don't have the time. And what he did is he measured it. Bose measured the photosynthetic efficiency of the leaves of hydrilla to be between 5.8 and 7.4%. Today we know that hydrilla is a facultative C4 plant that shifts from C3 to C4. So here, if you're not aware, C3 and C4 there are different kinds of plants. C3 is a kind of plant, C4 is another kind of plant. And actually that malate use is a very unique feature in C4 plant. And now we know that photosynthetic efficiency is less than 4%, about 4.6% in C3 plants, whereas in C4 plants, which this number can reach about 6%. So this number that Jagadish measured 100 years ago using this incredibly homemade, ingenuine device is not far from what is known today. And if you look at the chapter very carefully, you will see that he was, he was so detailed. He take care of everything. It comes up, and so I want to stop here. I don't want to go into the details. I think what I have done is I have convinced you that Jagadish Chandra Bose, we know him as a physicist. He has done incredible work in microwave research. In fact, because of his work in microwave research, on the dark side of the moon, there is a crater named after him. It's called the Bose crater. The IEEE, the international organization, honored him about 10 years back, giving him recognition. It does not matter. We claim, why did he get the Nobel Prize? It doesn't matter whether you get Nobel Prize or not. I think if you have done some incredible work, you will get recognized. Christ College, where he studied, when Jagadish's 150th birthday, Christ College came to Bose Institute and said they want to put up a bust of Jagadish Chandra Bose there. You know whose bust is there? Charles Darwin. So beside Charles Darwin bust, there is a bust of Jagadish Chandra Bose in Christ College. You may have read that UK was thinking of, publishing, taking out pound bills with Jagadish Chandra Bose's face on it. So Jagadish Chandra Bose is well known for his pioneering work in, microwave research. He also did pioneering work in plant electrophysiology. But I don't think many people know or aware of the amount of work that he did in photosynthesis. And it is not just the amount of work, a quantitative way of looking at it, and in the process, identifying new things that probably he was also not aware of what he was doing. And he did this when he was 65 years old. And he did this 100 years ago. Thank you very much.